Hello dear students. So today we are going to see some very important calculations from the principle of inheritance and variation. So here we will discuss all the important formula which is usually asked in NEET. So one very common formula asked is about the types of gamete. They will give you a genotype and certain condition and you have to calculate the types of gamete. So most common type of gamete type question is asked in case of independent assortment, right? Independent assortment. So this is first condition we will see. Now, if there is independent assortment and then they will give you several genotypes. For example, let's say capital A, this or this. So let's say this type of genotype they will give and so on. Let's say these are the genotype given to you and you have to find out the types of gamete in case of independent assortment. So when there is independent assortment, then we use the general formula for the type of gamete as 2 to the power n, where n is the number of heterozygous pair. Heterozygous pair, right? So let's see the formula, apply it here. Now here if I ask you this first one, here if I ask you, tell me how many heterozygous pair are there. See, you can clearly see this is one heterozygous pair and this is the second heterozygous pair. So here 2 heterozygous pair, it means n is equals to 2 here. So 2 to the power 2 is equals to 4. So how many types of gamete here? 4 types of gamete. Now here if we see what is the number of heterozygous pair, so you can see 1, 2, 3. So 3. So n is equals to 3. So 2 to the power 3 is equals to 8. Now here you do by yourself. How many n is there? So n value, see, is this heterozygous? No, this is not heterozygous. Is this heterozygous? No. Is this heterozygous? Yes. So n is equals to 1 here. So 2 to the power 1 is 2. Now in this case, last one if we see, now see this is not a heterozygous, homozygous pair, homozygous pair, homozygous pair. So what is the value of n? N is equals to 0. So 2 to the power 0 is equals to 1. So here you get only one type of gamete. So this is the basic formula based on independent assortment. Now here some more points are asked related, like when you see monohybrid cross, right? So in monohybrid cross, they ask that in monohybrid cross, how many types of gamete? Types of gamete in F1 generation. How many types of gamete in F1 generation? Now we know that in monohybrid cross, F1 generation is capital T small t. So you tell how many types of gamete? So here, of course, it is 1 heterozygous. So 2 to the power 1, that is 2 types of gamete. Now in dihybrid, if question is asked, in dihybrid cross, how many types of gamete in formed by the F1 generation? Right. So now in F1 generation, what is the genotype? Capital R, small r, capital Y, small y. So here the value of n is 2. So 2 to the power 2 is 4. So this is very common question asked about monohybrid and dihybrid. So these are very common question asked about the types of gamete. Now as I said, most of the question are asked from this category, independent assortment. But two another categories are there. Types of gamete in case of complete linkage, in case of complete linkage, and in case of incomplete linkage. Incomplete linkage. Let's say if these are the conditions. So first one we have seen independent assortment, second condition is complete linkage, third condition is incomplete linkage. Now if there is a complete linkage, now let's say see the same example, capital A, capital B small. Now if I say A and B both are linked, if I say both are linked, then how you will say, do the calculation? See, here also you use the formula 2 to the power n, but the only condition is the gene which are linked will be counted as 1. For example, here A and B is linked, so I will count them not as 2 pair, I will only count them as 1 pair. So here what we will say, n value is 1, so 2 to the power 1 is equals to 2. Understand, the pair which is linked, it will be counted as 1, right? Because they cannot be separated, so they are counted 1. So types of gamete is 2. Now let's see here, capital A. Now let's say this is 3 genes are there here. Now I give you this, that A and B here are linked, completely linked. Now how will you calculate? So as I said, here the value of n will be, see this is 1 heterozygous, but this we will count as together because they are linked, so we will count them as 1. So this is 1 and this is 2. So n value will be 2 here. 2 to the power 2 is equals to 4 and so on, right? Let's say this is the case. Now here if I say A and B are linked and C and D are linked, right? A and B are linked together and C and D are linked together. Now in such case, if question is asked, then how will you answer? So here what will be the value of n? See, the one which is linked, we will count together them as 1. This is linked, so I will also count it. So 1 and 2. So total 1 and 2. So the value of n is equals to 2. N is equals to 2. So 2 to the power 2 is equals to 4. So this is the case of complete linkage. Simple. Now coming to incomplete linkage. In this case, simple. Here the types of gamete is same as independent assortment. Same as independent assortment. Because all are possible. So here we will say types of gamete will be 4. In this case, even if they are incomplete linkage, but still the types of gamete will be following the simple 2 to the power n formula, and here 2 to the power 4, right? So it will be 16. So remember this, right? In incomplete linkage, types of gamete same as the independent assortment. Complete linkage, they are different. Now here in independent assortment, if you see, the types of gamete will always have 1:1, means all gametes will be in equal proportion, right? All gametes are always in equal proportion. But here in case of incomplete linkage, gamete types of gamete are same, but they are in unequal proportion. Why is it so? Because in case of incomplete linkage, the parental is greater than recombinant, right? So this is all about the types of gamete calculation. Now if we see, if the question is asked types of genotype, so here the general formula to calculate the types of genotype is 3 to the power n. If I say monohybrid cross, right? If I say F2 generation of monohybrid cross, what will be the types of gamete? So for monohybrid cross, value of n is 1. So it will be 3 to the power 1. So it is 3. We know that the ratio is 1:2:1. So it is 3. Then in dihybrid cross, right, the F2 generation, n is equals to 2. So 3 to the power 2, and here it will be 9, right? In trihybrid cross, in F2 generation, n is equals to 3. So 3 to the power 3 is 27. So this is simple types of genotype you can calculate, right? Now next, if we move to the types of phenotype. So in this type of phenotype, if we see, so again here the general formula used for type of phenotype is 2 to the power n, and here you will say monohybrid cross. Now everywhere it is for F2 generation. So in F2 generation it will be 2 to the power, for monohybrid it is 1, so it will be 2. For a dihybrid in F2 it is 2 to the power 2, so it is 4. For trihybrid it will be 2 to the power 3, so it will be 8. So in this way you can easily tell for types of phenotype for each of the types. For example, here we say the phenotypic ratio 3:1, and here commonly 9:3:1. These are the phenotypic ratio of monohybrid and dihybrid cross. So this is one very simple types of question asked with respect to F2 generation. Now for the calculation of numbers of genotype form. So here see, the number of genotypes is basically very simple formula. So here the formula is types of male gametes into types of female gamete. So this is the very simple formula, right? And if we apply it to monohybrid cross, so we know that in monohybrid cross, male and female both form how many types of gamete in the F1 generation? So you see male form 2 types of gamete, female form 2 types of gamete. So number of zygote form is 4. In dihybrid cross, if we see, so now we are talking about if gamete formed by F1 generation. So here 4, here also 4. So 4 into 4, 16. Then in trihybrid cross, in F1 generation, so we know that it is 8 and 8, so it will be 64. So this is how we calculate the number of zygote in monohybrid, dihybrid and trihybrid cross. Now here let's say randomly if they give you certain genotype and then they ask you types of or number of zygote. For example, let's say this A, small a, capital B, small b, crossed with capital A, small b, small a. Now here if you are asked that calculate the number of zygote formed in the offspring. So here see, let anyone you consider male, anyone you consider as female. So let's say this is male. So here how many types of gamete will be formed? So 2 heterozygous pair, so 4 types of gamete. And here how many types of gamete? Only 1, so 2 types of gamete. So 4 into 2, so total 8 zygotes are formed here, right? So easily you can calculate the number of zygote formed if the genotype is given. Now coming to a very important part where simple monohybrid ratios are asked. So see here it's very simple, so quickly I will go through it. See, monohybrid ratio, the basic question asked is first, what is the phenotypic ratio? Phenotypic ratio. So you say phenotypic ratio is 3:1. Genotypic ratio. So genotypic ratio is 1:2:1, right? 1:2:1. Then if I ask, and these all are in F2 generation I am talking about, right? Now if I say in F2 generation, tell me the parental phenotype. See, in F2 generation, if I say parental phenotype, so we say all are parental. So we say 100% phenotype is parental type. If I say the recombinant phenotype, so if parental is 100, so recombinant phenotype will be 0%. Similarly here if I say genotype, now we are focusing on parental genotype. So quickly let me write the genotype and then you will easily understand it. See, we say capital T, ratio capital T, small t, ratio small t, small t. Now these two are parental, isn't it? So 1, 2, 1. So what we say? This one and this one both are parental, so 1 plus 1. So can we say 2/4 are parental and 2/4 is 50%. So can we say 50% is parental genotype? And if I say recombinant genotype, so this one is recombinant genotype. So again 2/4, so 50% is recombinant genotype. This is about the monohybrid cross, right? So simple, these all are very simple part. Now let's go one step ahead here. Now let's say question is asked again about the F2 generation, because generally question is asked from the F2 generation. If I say in F2 generation, tell me the percentage of homozygous. So you will say 50%, right? You know that TT and small t are homozygous, so this is 50%. Then if I say percentage of heterozygous, so heterozygous, capital T, small t, so this is again 50%. Then if I ask you percentage of dominant phenotype, so dominant phenotype is always 3/4. You know no? That tall and dwarf. What is the ratio? 3:1. So it is 3/4. So can we say 75%? And if I say percentage of recessive phenotype, so it is 1/4, and that is 25%. Now on the basis of these percentage, sometimes they ask you number based question also. For example, let's say such question. They will say F2 has 2000 offspring, and then they will ask you how many of it will be homozygous, how many of it will be heterozygous, how many will have dominant phenotype, how many will have recessive phenotype. So you can easily calculate it on the basis of this number also. So this is all kind of question which can be possible from monohybrid cross. Now moving to dihybrid cross. So again, you know that in dihybrid cross, the phenotypic ratio is phenotypic ratio is 9 is to 3 is to 3 is to 1. And how many types of genotype? So 9 type of genotype, and genotypic ratio will be, see, the ratio depend on the order of genotype. That's why the ratio is different in different books. It doesn't mean it is given wrong. Just only thing you remember that in that ratio, for example, let's say 1 ratio, 2 ratio, 1 ratio, 2 ratio, 4 ratio, 1 ratio, 2 ratio, 1, right? So here what we are seeing, that 1, 2, 3, okay, no, so it 1, 2 I am missing, 2 ratio, 2 ratio 1. So this is, now this ratio, now you will see that this ratio may be a little bit of variable in different books, right? Now why it is variable? Because it depends on in which order I am writing the genotype. Only thing you have to notice that 1 will come 4 times, 1, 2, 3, 4, right? Generally in NEET you will never get to they will never ask you what is the ratio, right? Because ratio, for that they have to give the order of genotype, and there is no standard order of writing a genotype, right? So just remember this, that 1 will come 4 times, 2 will also come 4 times, and 4 will come 1 time. So this is the genotypic ratio. Now here, same way, if I ask you, out of this, if I ask you parental phenotype, now we know that this 9 and this 1, it is the parental phenotype. So we say 10 by 16 is parental phenotype. If I say recombinant phenotype, so it is 6 by 16. Same way, if I say parental genotype, so here this one 1, 1, so parental will be 2 by 16. And what will be the recombinant genotype? Rest all are recombinant genotype. So 14 by 16 will be the recombinant genotype. Parental genotype will be capital R, capital R, capital Y, and small r, small r, small y, small y. This is the parental genotype. So this is basic question. Sometimes they also ask that what is the ratio of the recombinant phenotype? See, this is the recombinant phenotype. So the ratio of recombinant phenotype is 3 is to 3. What is the ratio of parental phenotype? So 9 is to 1. So in this way also you remember, parental phenotypic ratio is 9 is to 1, recombinant is 3 is to 3, that is 1 is to 1, you can remember. Now moving ahead, in dihybrid, one more very interesting and important question is asked, that let's say if I give you a particular genotype, right, and if I ask you what is the fraction of these genotype, right? So see, there are total 9 type of genotype. No need to remember the 9 types of genotype, because in question you will be given the genotype and you will be asked its frequency. Now frequency also you don't have to remember. A very simple formula is there through which you can easily calculate the frequency. So for example, let's say this is the genotype given. Now how you will calculate the frequency? So if you see the ratio wise, this come 4. So 4 by 16. Right? Now how to say it without memorizing? See, here again you will follow the formula 2 to the power n. Very common formula used here in this chapter. N is the number of heterozygous pair. See, 1 pair, 2 pair. So 2 to the power 2, so 4 by 16. Now if I ask you this, tell me by yourself, what will be the frequency of this? So because this is also homozygous, this is also homozygous. So heterozygous pair is 0. So 2 to the power 0 by 16. That is 1 by 16. Similarly, if I ask you this, tell me what will be the fraction of this? So here 1 heterozygous. So you will write 2 by 16. Similarly, this, so you will write 2 by 16. Then if I ask this, then again you will say it will be 2 by 16. So in overall, what you will see, that you will realize that different genotype, there will be 3 values only, either 1 by 16, either 2 by 16, or either 4 by 16. 4 by 16 come once, 2 by 16 come 4 times, and 1 by 16 come 4 times. This is the case when there is one homozygous, when there is both homozygous, right? Here one homozygous, one heterozygous, and here both. So let me write it here, both heterozygous. This one is one homozygous, another heterozygous, and here both homozygous. So now on the basis of this, if question is asked, in F2 generation, how many genotype are both homozygous? So you will see the answer will be 4 by 16. In F2 generation, how many is one homozygous, one heterozygous? So you will write 2 by 16 into 4, so it will be 1 by, right, so it will be 1 by 2. Here also it will be both heterozygous, so 4 by 16. So 3 cases can be there in F2 generation, both homozygous, one homozygous, another heterozygous, or both heterozygous. So in this way, using this formula, you can easily calculate. So this is all about the dihybrid cross. Now if we say test cross, so test cross, there is again we will divide it into 2 parts. One is the monohybrid test cross, another is dihybrid test cross. So when we say monohybrid test cross, so in monohybrid test cross, the phenotypic ratio is 1:1, and genotypic ratio also is 1:1, right? How we represent a monohybrid cross? Capital T small t into small t, where this one is unknown genotype and this one is homozygous recessive. Now in monohybrid cross, if there is a in dihybrid cross, first let's see dihybrid cross. In dihybrid cross, the phenotypic ratio will be 1:1, and the genotypic ratio will be again 1:1, right? And here we represent it like this. One will be this, another will be this. Means one is unknown phenotype and another is the recessive genotype. Now here, if there is a complete linkage, sometime question is asked that in a dihybrid test cross, if there is a complete linkage, then what will be the phenotypic ratio? So phenotypic ratio in this case will be, yes, phenotypic ratio will be 1:1, and genotypic ratio will be again 1:1. So remember this, that in a dihybrid cross, if the condition is of complete linkage, then the phenotypic and genotypic ratio is same as the monohybrid cross. So you can see this ratio and this ratio is the same. So this is the test cross. Now another important point sometime asked is, if there is a test cross, let's say this, right, then what will be the frequency of this? Only these two are possible. So we know that they are in which ratio? 1:1. So this is also 1/2 and this is also 1/2. So each one is 50-50, right? So this is a simple test cross ratio. So through this you can easily calculate. Then coming to multiple allelism. So first of all, multiple allelism, how many gene control this? So it is controlled by how many gene? It is controlled by one gene, right? And this one gene has more than 2 allele. This is called multiple allelism. Now ABO blood group is an example of multiple allelism. Now ABO blood group, very common question sometime asked is, how many gene control ABO blood group? So it is one gene. And what is the name of the gene? The name of the gene is called I gene. And this I gene has how many allele? So it has IA, IB and IO. Clear? So here again, if question is asked, how many phenotype is there, right? How many phenotype and how many genotype is there? So very simple. Here if you see the genotype, so genotype is 6, right? And phenotype is how many types of blood group? So either blood group is A, either blood group is B, or it is AB, or it is O. So there are 4 types of phenotype. Now we know that A phenotype will have 2 types of genotype, either it is AA or AO. Similarly, B will be either BB or BO. AB only one, that is AB, and O also only one, that is OO. So you can see 1 genotype, 2, 3, 4, 5 and 6 genotype, right? So here you can easily find out. Now here one very important thing is, here IA and IB, they are codominant, right? So they are codominant. Now here blood group related questions are very important, right? So practice that blood group related question. Further here, let's say if the question here is asked that multiple allelism, so if I say ABO blood group, so can we say ABO blood group is an example of, can we say it is an example of multiple allelism? Yes, it is given. So it is example of multiple allelism. Now can we say that it is an example of codominance? Yes, it is an example of codominance. Then can we say it is an example of dominance? Yes, it is an example of dominance. If A is with O, B is with O, it is an example of dominance, codominance. When A is with B, it is codominance, and because it has A, B, O, so it is multiple allelism. So remember, here in blood group related question, a lot of statement type question is asked. For example, if I say AB blood group is always heterozygous, true or false? AB blood group is always heterozygous. So answer is yes, they are always heterozygous. If I say O blood group, it is always homozygous, right? Always homozygous. So this is also correct. And if I say A or B, they can be either homozygous or it can be heterozygous. Heterozygous. Another very important part is because multiple allelism has more than 2 allele, but one person, one individual can only have 2 allele. That's why multiple allelism is only observed in population. You cannot observe it in a individual, right? So that also is very important. Keep it in mind. Now another very important and a little bit of confusing is the polygenic inheritance. So polygenic inheritance, what we say here, when one character when it is controlled by multiple gene, when it is controlled by multiple gene, it is called polygenic inheritance. Now it is also called as quantitative inheritance. Quantitative inheritance. Now another important point, does polygenic inheritance follow Mendel's law? So it not follow Mendel's law, right? Mendel's law. Then another very important point, it depends on or it is affected by environment. Environment, right? Then further you can say it show additive effect. It show additive effect. So these are some simple common characters are there. When you read NCERT, all points are given. Now here, if question is asked about phenotype, that what are the different types of phenotypes? So here the formula is 2n plus 1, right? So for example, if I ask you, polygenic inheritance is controlled by 3 genes, right? How many types of phenotype? So if it is 3 genes, then the phenotype will be, put the formula and value. So it is 2 into 3 plus 1, so it is 7, right? Similarly, now genotype. So genotype formula is exactly the same. Genotype formula is 3 to the power n. So here the genotype will be, because it is 3 genes, so it will be 3 to the power 3, right? So types of genotype will be 27. So this part is clear. Now here some example is given in NCERT, and one example, very important example, is human skin color. Human skin color. Now human skin color is controlled by how many genes? So it is controlled by 3 genes, and in NCERT they have given A, B, C. Now so the one which is darkest skin will have all the dominant allele, right? This one will be the darkest, and the one which has all recessive, this will be the lightest, right? Now these are the two. Then in this way there will be, and now the complexion will depend, or the skin color will depend on the number of dominant allele. For example, this from here, let's start. Capital A, small b, small b, right? And in this way, as you keep on increasing the dominant allele, the phenotype will keep on changing. The phenotype will keep on changing like this. And finally, this one. Okay. So this one already done. So now this. So you can see these are the 7. These are the 7 phenotypes. Now here, if I say that which is the most common, which will be the most common? So the one which is intermediate. The one which is intermediate will be most common. So this is the intermediate and this is the most common one, right? So remember, so here darkest is 1, then 6, then 15, then 20, 15, 6, 1. So this you can also consider it as a ratio, that is 1 ratio, 6 ratio, 15 ratio, 20 ratio, 15 ratio 6 ratio 1, where this is the intermediate, right? And these are the two extreme phenotype. And if we plot the graph, it comes something like this. See? So this is the intermediate one, which is the maximum. We say it as 20, and then so on, 1, 6, 15, like this. Right? But yeah, these are not very much important as per the NCERT, but just keep it in mind because this is explained in detail about the skin color. So accordingly, you should answer it. So this was it, all the important points. Then rest of the points are the theory part. So just go through it, solve a lot of questions from here, and if there is any doubt, you can connect me. So best of luck. Keep on studying.